Hello and welcome to Sam Biology class. Friends, today we shall discuss about gene interaction that results in novel phenotype. We will focus primarily on the interaction between two independently asserting genes that produce a phenotype which is different from the one expressed by either of the parents. Because two genes interact or collaborate to produce the new phenotype, such genes are called collaborator genes and the new phenotype is known as novel phenotype. Some of the first evidence that a trait can be influenced by more than one gene was obtained by Batson and Punnett. From breeding experiments with chickens. Domestic breeds of chickens have different comb shapes. Combs are fleshy red-colored outgrowth present on the top of chicken's head. Wender's varieties of chickens have rose comb, Brahmas have pea combs, and leghorns have single combs. Batson and Punnett discovered that comb types is determined by two genes. One is rose comb gene and the other one is pea comb gene. Presence of rose comb gene is represented by uppercase R and its absence is represented by lowercase r. Similarly, presence of pea comb gene is represented by uppercase P and its absence by lowercase p. Both rose comb and pea comb gene express themselves in heterozygous state. So they are considered as dominant genes. Presence of at least one rose comb gene and absence of pea comb gene result in rose comb. Similarly, absence of rose comb gene and presence of at least one copy of pea comb gene result in pea comb. But when both the genes are absent, then the phenotype is single comb. But when both genes have at least one copy, then the resultant phenotype is walnut comb. Walnut comb is the novel phenotype which developed due to the interaction between rose comb gene and pea comb gene. When Batson and Punnett crossed rose comb Wenders with pea comb Brahmans, all F1 chickens had yet another comb type known as walnut comb. As previously mentioned, walnut comb is the novel phenotype which developed due to the interaction between rose comb gene and pea comb gene. When F1 chickens were intercrossed with each other, 4 types of progeny were obtained in the ratio 9:3:3:1. Out of 16, 9 chickens had walnut combs, 3 had rose combs, remaining 3 had pea combs, and only 1 had single combs. Analysis of F2 results indicate that walnut comb develop when both genes have at least one copy. These are the four genotypes that result in walnut comb. In all of them, both genes have at least one copy. Another phenotype that is rose comb developed when P-comb gene is absent but the rose comb gene have at least one copy. These are the genotypes that result in rose comb. And P-comb develops when rose comb gene is absent and P-comb genes have at least one copy. These are the genotypes that result in P-comb. But when both the genes are absent, then the result is single comb. In this example, walnut comb is the novel phenotype which developed due to the interaction between two independently asserting genes R and P. Chickens with walnut combs do not breed true for walnut combs. Instead, when a bird with walnut comb is crossed with another bird also with walnut comb, offsprings with other comb types are also produced. In this example, interaction takes place between rose comb gene and pea comb gene. Each gene has its own effect. Rose comb gene produces rose comb, whereas pea comb gene produces pea comb. But when the two genes are brought together, a new phenotype or novel phenotype develops. Walnut comb is the novel phenotype. Because rose comb gene collaborate with pea comb gene to produce walnut comb, these two genes are called collaborator genes. This experiment of Batson and Punnett demonstrated that a trait can be influenced by more than one gene. That means gene interaction takes place. So friends, this is all about gene interaction that result in novel phenotype. For more updates, subscribe my YouTube channel, Sam Biology Class.